Hello friends, welcome to the lecture series on basics of cell biology. Today we are going to learn about a new cellular organelles which is present in both prokaryotic cell as well as eukaryotic cell, that is nothing but the ribosomes. So let us see what are the ribosomes, what is its structure, chemical composition and functions. Ribosomes, they are very important cell organelles and they are actively involved in the process of protein synthesis. They are actually small, sub-microscopic, dense, rounded and granular particles. They are made up of from ribonucleoproteins. Ribosomes are also present in the mitochondria and chloroplast. They are present in both eukaryotic and prokaryotic cells. In prokaryotic cells, the ribosomes either occur freely in the cytoplasm, but in eukaryotic cell they occur in the cytoplasmic matrix freely as well as sometimes attached to outer membranes of endoplasmic reticulum and nucleus. A large number of ribosomes can be seen in cancerous cells and in the cells which are actively involved in the secretion of different types of hormones. For example, pancreatic cells, liver cells, plasma cells, thyroid cells and mammary gland cells. So ribosomes are present abundantly in these type of cells. Now ribosomes, they are for the first time discovered by the scientist Cloud in 1941 and he coined them at that time as microsomes. They are first observed in plant cells by Robinson and Brown in 1953. Afterwards, they have been reported in animal cells by Faraday in 1955. Ribosomes, they are 150 to 200 angstrom in diameter. They occur distributed singly but sometimes seen attached with mRNA, which are called as polysomes or polyribosomes. They are the sites of protein synthesis in the cell, hence they are known as protein factories of the cells. Amino acids are assembled in a specific sequence for the production of polypeptide chain. This is the function of ribosomes, hence they are known as engines, because all these ribosomes, they assemble different types of components for the process of protein synthesis. So they are known as engines or protein factories. There are different types of ribosomes and they can categorized into different types on the basis of sedimentation coefficient. Sedimentation coefficient, it is nothing but it is the speed at which the ribosomes settle down by centrifugation. It is expressed in the Svedberg unit. For example, it is known as S unit. The S is related with the size and molecular weight of the ribosomal particles, and on the basis of sedimentation coefficient, they are of two types. First type, it is 70S ribosomes and second type is of 80S ribosomes. Now first of all we will see what are the 70S ribosomes. These are the ribosomes, or they are also known as prokaryotic ribosomes. They are having sedimentation coefficient 70S and the molecular weight is 2.7 into 10 raised to 6 Daltons. According to the electron microscope studies, they are dry ribosomes. The dry ribosomes are 170 into 170 into 200 angstrom in dimension. These types of ribosomes occur in prokaryotic cell such as blue-green algal cells and bacterial cells. Also, they are present in mitochondria and chloroplast of eukaryotic cells. They are made up of from two subunits, that is 50S and 30S. So here you can see this is the structure of ribosomes and here 70S ribosome have been shown. So this is known as 30S subunit, it is smaller subunit, and this is 50S subunit, it is larger subunit. Both these subunits, they will come together when there is 0.0 0.01 molar magnesium ion concentration is there, then they will be associated with each other. But when the molar concentration of Mg++ ion it becomes less than 0.001 molar, then they will become dissociated like this. So these are 70S ribosomes. Now we will see second type, that is 80S ribosomes. The 80S ribosomes are present only in the cytoplasm of eukaryotic cells and they are absent in prokaryotic cell. They are abundantly present in plants as well as animal cells and they are having sedimentation coefficient 80S. Molecular weight of 80S ribosomes, it is 40 into 10 raised to 6 Daltons. So these 80S ribosomes, they are made up of from two subunits like 60S and 40S. And these 80S ribosomes, they are comparatively larger than that of 70S ribosomes. Here two subunits have been shown, 40S subunit and 60S subunit. This is smaller subunit and this is larger subunit. These two subunits, they will associate in the higher concentration, that is 0.001 molar magnesium ion concentration, and they again get dissociated when molar concentration of Mg++ ion it becomes less than 0.001 molar. So these are nothing but the 80S ribosomes. Now we will proceed to see structure of ribosomes. Here you can see this is the actual structure of ribosomes. Ribosomes, they are spherical in shape and they are 150 to 250 angstrom in diameter. Each ribosome, it is porous, hydrated and composed of two subunits, namely larger subunit and smaller subunit. Here both subunits have been shown. This is smaller subunit and this is larger subunit. The larger subunit is always dome shaped and smaller subunit is always a cap like structure. So this is dome shaped larger subunit and this is the smaller cap like subunit. Smaller subunit is present above larger subunit. So here you can see this is smaller subunit which is present above the larger subunit. The 70S ribosomes, it is made up of two subunits like 50S and 30S. So 70S ribosome, it contains two subunits. First is larger 50S subunit and second is 30S subunit which is smaller subunit. If you see 80S ribosomes, they consist of two subunits, that is larger dome shaped 60S subunit and smaller cap like 40S subunits. Here you can see both subunits have been shown here. In 80S ribosomes, there are two subunits, 40S and 60S, and they forms 80S ribosomes. Here you can see this is dome shaped smaller subunit and this is this is dome shaped larger subunit and this is cap like smaller subunit. Here you can see both the subunits of 70S and 80S ribosomes, they is separated by a narrow cleft. So this is nothing but a narrow cleft. Both subunits, that is this smaller subunit and larger subunit, both are separated from each other by a narrow cleft. So this is nothing but a narrow cleft. The two ribosomal subunits remains united with each other due to high concentration of Mg++, that is 0.001 molar ions, as we have discussed in earlier slide. Here you can see both these units, they remain together when molar concentration of Mg, that is magnesium ions, it is higher than that of 0.001 molar. But they get dissociated, they get separated when concentration of magnesium ions it becomes less than 0.001 molar. When the concentration of Mg++ ion it reduces in the matrix, both ribosomal subunits get separated. At high concentration, as we have seen, of Mg, that is magnesium ions in the matrix, the two ribosomes or ribosomal units becomes associated with each other and they are known as dimer. So this part, it is known as dimer. So why it is known as dimer? Because both subunits come together to form a structure like a dimer. During the process of protein synthesis, many ribosomes are aggregated due to common messenger RNA and they form a structure. This structure is known as polyribosome, or it is also known as polysome. So here you can see all different ribosomes come together and they are arranged on a common messenger RNA and they form a structure that is polyribosome. Here 5 ribosomes, they are shown arranged on this mRNA strand and they have formed the structure of polyribosome, or it is also known as polysome. Now we will proceed for chemical composition of ribosomes. As we know, RNA and proteins are the major constituents of ribosomes. The 70S ribosomes, they contains more RNA, that is 60 to 40% than proteins. So see here, 70S ribosome, they contains more RNA and less proteins. But in 80S ribosomes, you can see more concentration of proteins and less concentration of RNA. So here it has been shown that the 70S ribosome, they contain 60 to 40% of RNA and 36 to 37% of protein. But 80S ribosome, they contains 56 to 60% of proteins and 40 to 46% of RNA. Lipids are not present into the ribosomes. Now we will see one by one chemicals which are present into the ribosome. The first and most important chemical which is present in ribosome, it is ribosomal RNA. 3 different types of rRNA are present in 70S ribosomes, which include 23S rRNA, 16S rRNA and 5S rRNA. Larger subunit contains 23S rRNA and 5S rRNA, while 16S rRNA are present in smaller subunit of 70S ribosomes. In 80S subunit or 80S ribosomes, so here should be ribosome, 80S ribosome, there are 4 types of rRNA: 20S rRNA, 18S rRNA, 5S rRNA and 5.8S rRNA. Larger, that is 60S subunit, it contains 20S rRNA, 5S rRNA and 5.8S rRNA, while smaller, that is 40S subunit, it contains only 18S rRNA. So these are different types of rRNA which are present, means ribosomal RNA, which are present in both 70S and 80S ribosomes. Now, second most important constituent of ribosome, it is ribosomal proteins. In 70S ribosomes, if you see of E. coli, because E. coli, it is a bacteria and it has prokaryotic cell, and we know in bacteria and in prokaryotic cells 70S ribosomes are present. So these 70S ribosomes of E. coli, they contains 55 ribosomal proteins, and all these 55 ribosomal protein, they have been recorded from both the subunits of the 70S ribosome, that is 50S subunit and 30S subunits. Larger 50S subunit contains about 32 to 34 proteins, and smaller, that is 30S subunit of 70S ribosome, it contains about 21 proteins. Now we will see what about 80S ribosomes. 80S ribosomes of eukaryotes, they contains about 80 proteins, out of which 30 proteins have been reported from smaller, that is 40S subunit, and 50 proteins are isolated from the larger, that is 60S subunit. Now last chemical component which is present in ribosome, it is metallic ions. So ribosomes of both, that is prokaryotes and eukaryotes, means ribosomes of both types 70s and 80s, they contains divalent low molecular weight metallic ions like magnesium, calcium, manganese, and they plays very important role in the process of functioning of the ribosomes. Now in last slide we will see what are the functions of ribosomes. Ribosomes are very important to play the function of protein synthesis, as we have discussed. Protein is made up of a long and linear polypeptide chain. We know it is made up of from amino acids which are linked to each other by peptide bonds. Ribosomes are important engines, as we have seen. They are the factories to produce proteins in cytoplasm, mitochondria, and chloroplast. Next function of ribosomes, it is coordination. Ribosome, they are very important machinery. They will assemble different components required for the protein synthesis. They include tRNA, amino acids, mRNA, and so that they are known as the protein factories. And as they coordinate all these components required for the protein synthesis, they play very important role of coordination. Now last function of ribosomes, it is protection. Ribosomes play very important function of protection of both, that is RNA as well as newly synthesized protein molecule. How? We will see. Ribosomes protect mRNA strand passing between the two subunits of each ribosomes from the action of enzyme nuclease. So we know nucleases are the enzymes which are present into the cell, and these nucleases, they cause the degradation and digestion of RNA as well as DNA. In the process of protein synthesis, mRNA plays very important role. So these nucleases which are present into the cell, they can break down mRNA, but ribosomes, they protect these mRNA by their two subunits, and they protect such type of mRNA from the enzyme nucleases, and they are not get breakdown. So also there is another nascent polypeptide chain of protein molecule, which is newly synthesized protein, which is passing through the tunnel of large subunit. It get protected, or it is also protected by ribosomes from the protein digesting enzyme, that is proteases, because these nucleases and proteases both are present into the cell cytoplasm, and they can break down or digest RNA as well as protein. So ribosomes play a very important role of protection of this mRNA as well as protein from these two enzymes, that is nucleases and proteases. So all these are the functions of ribosomes. Hope you understand the concept of ribosomes. Thanks for your attention. If you like video, you can share with your friends, like, comment, and subscribe the channel. Thanks for your attention.